Good evening and welcome to the Canadian Hemophilia Society's second webinar on gene therapy. My name is Sarah Ford and I am the Chief Executive Officer of the Canadian Hemophilia Society. As part of the CHS Gene Therapy Education Program, we're pleased to offer many educational tools, including this webinar series, for our community to become better informed about this novel therapy option. You can find all this information on our website, and the address can be found at the end of this webinar. I'd also like to quickly mention that this webinar is currently being recorded and will be available on the CHS website as soon as possible. Tonight's program will feature presentations by Dr. Roy KHALIFÉ, followed by David Page. And I'd like to remind you that you can submit your questions to either of the speakers during the webinar by using the Q&A function at the bottom of your screen. So to begin, I would like to introduce Dr. Roy KHALIFÉ. Dr. KHALIFÉ is a hematologist at the Ottawa Hospital and co-director of the Ottawa Regional Bleeding Disorders program. He's an assistant professor at the University of Ottawa, and his research interests focus on the implementation science in medicine and medical education to optimize patient care and outcomes. So welcome, Dr. KHALIFÉ, and thank you very much for joining us this evening, and I will turn this over to you.
Thank you, Sarah, and thank you to the CHS for inviting me to give this presentation. I'm hoping that you can see my slides now. Yes, I'm going to assume yes. So welcome, everyone. Delighted to talk about hemophilia gene therapies today. So I'm Roy KHALIFÉ. I'm a hematologist in Ottawa, and do have I think conflicts with everybody who works in the bleeding disorder world. So I'll leave this here for a few seconds. So starting off, just to get everybody on the same page in terms of gene therapy, you probably have heard a lot of it about it, and you will keep hearing about it. I think this is very innovative and transformative therapy that is happening in hemophilia and many other hematologic conditions that we manage. So gene therapy, really what it does, what we're trying to do is to take to introduce a functional gene, so something that's going to correct either correct or replace a faulty gene, so it's something that doesn't work. And the concept of gene therapy has been around for many decades. In fact, probably emerged in the 70s, but failed the first trials with initial viral vectors or transport system. But the idea remained attractive. And so hemophilia became a sort of a prototypic disease or condition for gene therapy. And the goal here for hemophilia would be to reduce or eliminate the need for regular infusions of clotting factors by targeting the root cause of hemophilia and the production of that missing protein, so factor 8 or factor 9, and offering a potential long-term solution. Now, lots of work is happening and happened in gene therapy with many different vectors or transport system where you're trying to transport the gene into a cell in order to be able to produce a protein. The adeno-associated virus, or AAV, is currently the preferred vectors for gene delivery, considering its safety and effectiveness. Lentiviral vectors are also being tested and can be used and can ensure long-term expression, but they do carry some additional risks, so I don't think we're there yet. The CRISPR system is also potentially has significant potential in hemophilia therapy as an effective tool for genome editing. And non-viral vectors, so nanoparticles or microvesicles, can also be used in hemophilia treatment, but they have low efficiency and potential some questionable safety of DNA delivery. So currently not in favor or limited use in terms of potential vectors. So really the focus for tonight and what's being presented is on gene therapies that use AAV as their platform. So AAV or adeno-associated virus are small viruses that can infect humans, but do not cause disease. They're not pathogenic. And so they are also more predictable in terms of their behavior, and we can more easily work with from a lab perspective compared to potentially other viruses. They do, or there is a potential where they can integrate in someone's genome when you're infusing it. But that rates of integration where the virus or the DNA integrates in someone's genetic coding genome is negligible. And several serotypes of AAV exist, and some of them have affinity to different organs. So you'll see different gene therapy products using different serotypes of these adeno-associated virus. And many people might have what we call neutralizing antibodies. So these are antibodies that people develop that are targeted against the adeno-associated virus. So in the context of a gene therapy, if you do have these pre-existing antibodies, it may potentially render the product less efficacious and potentially make you ineligible for certain products. And we'll talk more about that. So this is just to keep in mind. Now, how these products work, or how this gene therapy work, and I'm putting this in one slide, so summarizing decades of work and complexity into one slide. But essentially, what you're trying to do is infuse this gene therapy, which is a bunch of these genes integrated into the AAV virus, which is there to transport that gene into the liver cells, into the nucleus of your liver cells. And so that AAV will bind to your liver cell, get inside your liver cell through that process of endocytosis. Inside your liver cells, it will be delivered to the nucleus, and the gene product will be released from the capsid of that virus, and with the goal of starting to transcribe that DNA into the next step, which is the RNA, and that RNA will then be able to produce the protein. So in this case, factor 8 or factor 9. And that protein will then get released into your bloodstream to be able to do its role or effect, which is to help you clot when you need it, when you're bleeding, for example. So I'm going to touch on the products that are currently available and potentially ready for commercial use in Canada in the near future, but or already in commercial use in other countries. First product that I'm going to present is Roctavian. So I'm going to stick with the commercial names because, as you can see, the trade names are somewhat convoluted. So Roctavian is a factor 8 based gene, so for hemophilia A. And this is probably what started. So the first publication was in 2022, and it's currently approved and commercially available in the United States and also in European countries. However, in Canada, sadly, the company decided not to pursue commercialization in our country. So, whether this changes or not in the future, I can't say, but nevertheless, just some ideas about how it's working. From this phase 3 trial, what we see, which is looking at the efficacy and safety of this product. So they looked at it in 134 adult men with severe hemophilia A, so less than 1%, and with a single infusion, they were able to demonstrate a reduction in the annualized bleeding rate by about 84%. And reduction in factor 8 concentrate use by close to 100%, so 98%. Now, the median factor 8 activity, you see that it does increase after the infusion. But what's happening over time with this factor 8 product, we're seeing a decline. Now, decline is slow, and sort of stabilizing. But what this means in the future, we'll talk a little bit more about durability later on, but just to keep that in mind. This is the update from this trial, which is presenting 3 years outcomes following a one-time infusion of this product of Roctavian. And again, you're seeing annualized bleeding rate that are decreased by about 80% by this time, by 3 years, with a mean, so an average factor VIII activity that is sitting around 18% at year 3. And again, there's a range, obviously, for different people will respond, will have different responses, but this is the average that's been reported at a 3-year mark. Now, on the hemophilia B side, so we have 2 products. Both products are now Health Canada approved and are undergoing negotiation for reimbursement. So David will be talking more about that process later on. But Hemgenix is one of the products. So this is by CSL Behring, and this was tested in 54 men with severe, or what they also call moderately severe hemophilia B. So this is a different classification. So essentially, it's taking anybody with a 2% factor IX level and less. And they had people between the ages of 19 and 75 in that trial. And again, it's a single dose for the infusion of the gene therapy, with reduction in the annualized bleeding rate by 64%. And again, 96% of people discontinued factor IX prophylaxis. So here the caveat to that is, so this is 52 people out of 54. However, one person only received 10% of the dose. And so this person was the first one to get the gene therapy product at that institution, and they had a reaction, and the infusion was aborted. So of course they didn't have really the response that is expected. And then the other person who did not respond in that trial is someone who had very high levels or titers of the adeno-associated virus serotype 5 neutralizing antibodies. And so they did not show a response in that trial. So what's interesting with this product, so Hemgenix, is the company decided that whether you do have neutralizing antibodies against AAV5 or no antibodies, they were going to infuse, you would get the infusion regardless. And so they demonstrated that even if some people had the presence of these neutralizing antibodies, they would still be able to mount a response and potentially come off prophylaxis. So this is a potential advantage in terms of Hemgenix. So regardless of whether you have neutralizing antibodies, you could potentially respond. And the mean factor IX activity within that trial after 18 months was 36.8%. Now, the updates from this trial that were presented this year. So this is up to 2 years of data so far. And again, showing a persistent decrease in terms of bleeding rates, but also still that 96% of people remain off factor IX clotting factors concentrates with a mean factor IX that is sitting around 36.7%. Again, this is an average. So, and about a third of people that participated in that trial had levels over 40%, so in the non-hemophilia range. Now, what they also demonstrated is even if you had these positive antibodies, so the red line, they're still showing a response, a good response, so sitting again about 30-ish%. But if you don't have AAV5 neutralizing antibodies, your response perhaps is a little bit better. And so this is again for us reassuring to see that this is a possibility regardless of the presence of these neutralizing antibodies or not. Now, the other product that is currently Health Canada approved is Becvest. So Becvest is a product by Pfizer. So in this setting, so they had 45 men. Again, severe hemophilia B and moderately severe hemophilia B, which was defined as 2% or less. And with this specific product, to be enrolled in the trial, they had to have a negative test for neutralizing antibodies against the adeno-associated virus that was used in that for that product. And so again, here they're demonstrating a decrease in terms of your annualized bleeding rate, but by about 70%. And more than 80% of people in that trial were able to achieve levels in the mild hemophilia range with a mean or an average factor IX activity at about 27% by 2 years of data. So this trial was just published in the last year, and there'll be updates, over time from the company as well. Now, on the safety side, for all three products, overall, the gene therapy products are safe for use, and the main things that we see are related to the liver function. So liver enzymes elevation, toxicity to the liver, and you see, for example, in the factor VIII product, Roctavian, about 85% of people had increase in their liver enzymes. Now, that doesn't mean that we can't treat that or fix that, but it is nevertheless a potential side effects or toxicity that needs to be addressed. This is less problematic in the factor IX products, so Hemgenix and Becvest. So about 20% of people in Hemgenix, and then about almost close to 50% in those with Becvest. What this leads to, essentially, when you have this elevation in your liver enzymes, the idea is that the product is probably working. There is a response from your immune system where there's this inflammation that is happening in your liver, which may be a good thing and tell us that your body is responding to that. But at the same time, we don't want to let it go wild without treatment, because it can potentially affect your response and show a decrease over time in terms of your factor levels. So what happened? There's different protocols, and what happened if your liver enzymes increase after gene therapy. The treatment is using corticosteroids. So steroids are medications that are quite familiar to hematologists. We use them for many immune-related conditions that we manage in hematology, but that's not something that the hemophilia community is used to, because that's not a treatment in hemophilia. And so what you see here is that in the Roctavian trial, up to 80% of people needed steroids to target these liver enzymes, and most people started within 2 months after gene therapy infusion, and treatment duration was quite long, up to 230 days. In the Hemgenix trial, much less need for steroids in that trial, and again, generally within about 2-3 months. Sorry, this is the duration, about 80 days. The Becvest trial, we see a bit more or higher needs than the Hemgenix in terms of steroids, and again within about a month or so, and duration about 95 days. So this is something important to keep in mind, and you will be counseled about. We will be talking about that for anybody who's interested in going through gene therapy, to be prepared to think about the need for potential steroids and for how long. And the problem with that is that corticosteroids are very effective medications when we need them, in terms of tapering the immune system, putting some brakes on the immune system and the inflammation in the body. But they also have a lot of effects on the body, and especially when used for a long time, people can develop many health issues that are related to the corticosteroids use. So these are things to keep in mind and go through and discuss, and of course, with the aim of minimizing how much corticosteroids someone need, we always aim to use the shortest duration possible, because of the potential of all of these these effects on your body and changes. Now, what we still don't fully understand, and we may never potentially fully understand, is why some people respond better than others, and why we see this variability in terms of how factor 8 is as expression is versus factor 9, and between individuals. And there's many factors that are hypothesized to be involved, and potentially probably more than one factor at a time um, will be responsible for a response, or a partial response, or a lack of response for someone. And these factors will target different um, steps in terms of that delivery of the gene products. So things like the neutralizing antibodies against the AAV, to things like potentially your blood group, your von Willebrand factor, especially when we're talking about factor 8 or hemophilia A. Things like your immune system, and the potential use of immune suppressing medications. So lots of things to take into consideration in terms of when we see the different response rates between individuals. In terms of how long the gene therapy is expected to last, well, that's the million-dollar question. I don't think anybody's going to be able to answer that question, and no clinical trial is going to be able to answer that question within sort of that clinical trial setting. We can't necessarily wait 15, 20 years to get these answers. And so part of that process will happen after gene therapy gets into the clinics, and of course monitoring and long-term monitoring to be able to gather that data and figure out what's happening in terms of response and duration of response. But what we know so far, and these are based on the phase 1 or phase 2 trials, so that are looking at whether, for example, this gene therapy works, and so on. And so this is Roctavian, so again, the hemophilia A product. And what you see here, this is efficacy over 7 years. So again, they're showing that efficacy, but there is a decline over time. And what that decline will look like, over 10 years, 15 years is unknown yet, and whether people are going to have to go back to prophylaxis after 10, 15, 20 years. Again, that remains a question. On the factor 9 side, we have for hemogenics. So again, in the phase 2, up to 4 years of data, and again here showing that no patients in that trial actually returned to prophylaxis, and none of these patients required corticosteroids. So again, this is a small number of patients. And they had 2 different arms for a smaller dose versus a bigger dose. But you see that they're maintaining their levels around 40 to 50% after 4 years. Now in the in the Beckfest product, for that phase 2 trial, now data up to 6 years in terms of efficacy. And again, the factor 9 activity is maintained within that mild hemophilia range at about 22%, again, plus or minus 12%. And again, in that phase 2 trial, none of the participants resumed factor 9 prophylaxis. So overall, this is reassuring, but that remains a big question, because once you've received an AAV-based gene therapy, there is no turning back, and there is no possibility to get another AAV-based gene therapy, presumably, because your body's going to make these antibodies against AAV once they've been exposed to it. So moving along to who will be eligible to get gene therapy. So if we follow the indication, or at least by Health Canada approval for the hemophilia B products, Hemogenics and Beckfest, both of them in that context is going to be adult based, so over 18 years, and that's primarily because of how the trials were done. Would a 16-year-old or a 15-year-old or 14-year-old respond? Presumably. It's a question of when the liver has grown enough to be able to take on the gene therapy. And there is some talks and potentially doing trials in pediatric settings, but that will probably come in the future. Um, in terms of Health Canada approval for Hemogenics, so they didn't specify a threshold for factor 9 activity, just anybody who's on a routine prophylaxis. What is important to note is that nobody in the trial had factor 9 over 2%, but there is no reason to think that someone with a 3% or a 4% or a 5% who needs prophylaxis and who wants to potentially undergo gene therapy, there's no reason to think that they would not respond or they would not benefit just because they have 4% and not 2 or less percent. The PECVES, on the other hand, Health Canada was, I guess, stricter in terms of who it's indicated for, and they did mention 2% and less, and that people had to be negative for these neutralizing antibodies against the variant that was used in that trial, which is the AAV serotype RH74. So anybody who's undergoing gene therapy will be tested for these AAV. Now, as I mentioned, Roctavian is not Health Canada approved, and this was suspended indefinitely, based on the decision, the company's decision. But it would have been for severe hemophilia A, unless they decided otherwise. As I mentioned, anybody who's contemplating or who wants to undergo gene therapy will be tested for these neutralizing antibodies. In the context of Hemogenics, we are mandated to test. However, for most people, even if you are positive, you will still be eligible for gene therapy. The caveat to that in terms of the monograph is anybody who's above a predetermined threshold will not be able to get it. And what that predetermined threshold, I have not seen yet in terms of any numbers written down, but I might have missed it. Again, not having allo antibodies or inhibitors to factor 9, not having advanced liver disease, or of course active hepatitis B or C infection or uncontrolled HIV infection. Other things that we're also going to have to think about in terms of who might be a good candidate, having good social support to go through this process, understand that process, and be ready to go through it. If you are planning to have a family in the near future, there are reproductive concerns that have to be taken into consideration, and acceptance to potentially delay family planning for a year or more after a gene therapy infusion. And this is again based on Health Canada indications. Having mental health concerns, trying to deal with them prior to getting into gene therapy, because there is a lot of work that is going to happen afterwards in terms of monitoring, going to the lab, getting blood work done, potentially getting steroids, which can also impact someone's mental health, and being able to accept the intensity of the short-term monitoring, the ability to tolerate corticosteroids, so not having uncontrolled hypertension, for example, or high blood pressure, uncontrolled diabetes, which will worsen with the steroids treatment. Alcohol, so abstinence, well controlling, intake of alcohol, because any damage to the liver could potentially reduce the response, or eventually over time potentially diminish the response after gene therapy. Having realistic expectations, so going into it that, understanding that it might not potentially work, it might work for a bit, and you might need to go back on prophylaxis, although, as I said, the efficacy is pretty good. And accepting the long-term monitoring. This is we need to learn more and need to be able to collect that data over time and be part of that CBDR registry, so that everybody understands what's happening, what's going on after gene therapy. And that's a mandate with any of these products through Health Canada. And of course, giving that informed consent. Should sex play a role? If a woman has factor 9 of 2% or 1% or less, should she be eligible or not? Women were not part of the trial. Part of the clinical criteria actually excluded women, but we know that from a biological perspective, there's no reason why they would not respond. Now, if someone is family planning again within reproductive ages, it's very unlikely to be offered in that setting. Again, because of the unknown or uncertainties about the potential effects in terms of reproductive system. Now, in terms of geography, again, we live in a very big country. Geography should not theoretically make you ineligible for gene therapy, but we know that there is inequities with whoever live, close to a hemophilia treatment center versus someone who lives very far away. Well, that ability to potentially get it. Ideally, we want everybody who wants it to be able to get it and closer to home. We already also know that there is discrepancy within Canada in terms of approval and from a ministry perspective, government perspective, approval for gene therapy. And I know David will touch base on these decisions between Canada and Quebec in terms of gene therapy. So I did want to emphasize in terms of liver health, I think this is the most important and crucial point for everybody to focus on. It's critical to the success of gene therapy, but also minimizing toxicity. All this gene therapy that is being infused is going into your liver. The liver is going to be producing your factor 8 or factor 9. So we need to take care of that liver. There's going to be extensive evaluation of that liver health before getting into gene therapy, but also important lifestyle measures. So weight management, decreasing that amount of inflammation and fat deposits within the liver, attention to alcohol use, which will damage the liver, and any medication that could potentially damage, or be toxic to the liver as well. As I said, we live in a very big country, so this is we had conducted a survey within the AHCDC to try to figure out where we would be starting in terms of gene therapy infusions. These are some of the centers who may be ready as soon as we have approval or reimbursement. So in Edmonton, McMaster, who has participated in many of the trials, Toronto, and then Ottawa, we would be ready as well. In Quebec, I didn't have a formal answer, but maybe later Dr. Saint-Louis can give us some hints. But I think Montreal, and I've heard potentially Quebec City will be could potentially infuse. But never, all this to say is within all the clinical directors, in terms of the discussion, if there is someone in Vancouver, someone in Winnipeg, someone in New Brunswick who wants to undergo gene therapy, in our discussion, most of our centers are able and should be able to infuse it. Sometimes there's just some logistical aspect of things to go around from within the hospital, but there shouldn't be a reason why we wouldn't be able to offer it close to home. Some centers will be more or ready faster than others, but we're trying to make sure that every center who wants or is able to give gene therapy has the basic information to do so, and of course the education to be able to monitor afterwards and take care of any potential side effects. So lastly, to me, what is also important in someone who will be undergoing gene therapy is to be able to understand and handle the uncertainties related to the long-term consequences or effects of gene therapy. There's things that we know in terms of current efficacy, current potential short-term toxicities, but there's still a lot of things that we don't know or we think we know, but we don't really know. And I think this is a framework that was presented by Glenn Pierce and Dr. Alfonso Iorio a few years ago, and it's still true to this day. There's, and we're learning from other conditions that have undergone gene therapy. We're still learning and we'll continue to learn about this. But someone who is interested has to be able to stomach these unknowns before getting into it. There's not a lot of data about patient experiences that has been published. I just wanted to bring this study from the UK by Simon Fletcher that have looked at patient experiences who have undergone gene therapy. So they interviewed 16 individuals with hemophilia, so 11 with hemophilia A, 5 with hemophilia B, and 10 of their family members to really understand the motivations, experiences, and sort of outcomes after gene therapy. And what they presented is really in terms of motivations, that desire to help future generations was a key motivator, but also viewing gene therapy as a potential cure and life-changing opportunity. What I think is also important for us in terms of clinicians or treaters understanding the challenges that people experience during that treatment or during the therapy, and the immune suppression, so corticosteroids and the side effects were a major challenge. So just the experience in terms of physical and mental health issues, insomnia, depression, anger, but also the loss of autonomy, the constraints within the clinical trial protocols, the amount of blood work and clinic visits, it's a lot on someone, especially in the short term. So for us to understand, because this is going to translate into the clinical setting, even though it's not a clinical trial, we're still going to follow a lot of these protocols. So important for us to understand and counsel people about that. And I think that's a reality for a lot of things, the insufficient psychosocial support, that's from the UK, but I don't think we're any better in Canada in terms of addressing the mental health and psychological challenge that people experience. Lastly, the control, so that sense of liberation or after undergoing gene therapy, not having to do factor IX infusions, that reduction in terms of dependency, but also concerns about the long-term effect, the uncertainty. So that balance within that control. So interesting things that came out of that study, and I think we'll be learning more about these experiences and perspective as more people undergo these treatments. So I'm 2 minutes over. I apologize, but I think this gene therapy offers a lot of hope, but will come with challenges that we need to carefully address and prepare people for. And I think without say the long-term monitoring is crucial for us to keep an eye on and make sure that people are doing well and responding well. So with that, I will stop here and happy to take any questions.
Thank you very much, Dr. KHALIFÉ. We very much appreciate your contributions tonight and your presentation. A couple questions to start, and just as a reminder to the audience, you can put your questions in the Q&A, and if they're not addressed during this question and answer period, we can also address it following David's presentation too. So first question comes regarding the Roctavian experience that's happening outside of Canada. Do you have a rough idea of how many patients have been treated already for hemophilia A with Roctavian outside of Canada?
Yeah, I don't. To be honest, I know that Hemgenics, there's about 22 in the United States so far, as far as I know. The Roctavian, I didn't look at the most up-to-date numbers that has been reported. I'm not sure
Okay.
if Fred or Dr. Saint Louis might have an idea, but we
Yeah,
can.
and we can always look into
Yeah,
that also with David. Another question that we had is what happens if the gene therapy doesn't work? Meaning,
yeah.
at what period do you know that this is not feasible with regards to factor levels and that you have to readjust the treatment regimen?
Yeah. So this is a very interesting question, and it's a question that we actually got in terms of a community of hemophilia physicians in Canada to address as the provinces are negotiating the price of gene therapy, in the sense of, well, how would you define gene therapy failure? How would you define when someone returns to prophylaxis? And there isn't a definition and set in stone. And in terms of discussion within our groups, the decision is going to be very much individualized, right? So just because your level dropped back down to 5%, well, maybe you don't need to go back on prophylaxis. You can certainly live with that level, but with the understanding that you're still at risk of bleeding. And we would need to give you factor at some points, either for surgery or trauma or whatnot. Now, whether, to me, if you get gene therapy, you respond, and then your level go back down to where you were before gene therapy, that to me is a lack of response. And likely that you will require to go back on prophylaxis because you're down to very severe ranges and potentially, at risk of rebleeding and bleeding. And of course, if someone respond, but then goes down and you're sitting at a 5%, but you are bleeding and having recurrent bleeding issues. That's where that discussion comes in. We have to do something, right? We need to prevent these bleeds. So it's not a question, or it's not an answer that is set in stone. And I think it all depends on, every individual circumstances, in my opinion.
And I assume that's part of the reason why that network of the HCDC with all the treatment centers across Canada, even with the dosing happening in one center, I would assume that's part of your questions that you're discussing as how the follow-up is also occurring with the other treatment centers.
Correct. So what I didn't present today is that idea of what we call hub and spoke model, which is prevalent in Europe and the US, in terms of how someone who wants gene therapy will be referred to a hub, which will infuse the gene therapy, and then they go back to their HTC, their spoke. Um, and if you speak to anybody within Canada, there's a lot of different opinions, and we're all opinionated in a way, but that's a good thing because we can always bounce ideas and discuss these things. But we've always operated, each HTC has always operated in an independent fashion in a way, and we've always incorporated any new treatment in hemophilia across all HTCs. So in our discussion, there's no reason why one HTC should be seen as a hub and the other as a spoke, when we could all potentially offer that. And pretty much most of our HTCs are within tertiary care centers, academic centers that offer many other treatments that are way more out there and severe in terms of consequences and toxicity than gene therapy. So I think most of us are able to do that, and that's why that hub and spoke model may not be necessarily what we'll go to in Canada. But initially, there may be people who are referred to different centers depending on who's set up to start gene therapy. And the idea is that every HTC is well educated and ready to take on that patient as soon as they go back, because you're going to get the gene therapy and you're going to be able to go home, pretty much the next day, because the infusion is about an hour, and the monitoring really happens afterwards, and that's going to be close to home.
Okay. Thank you very much, Dr. KHALIFÉ. And with that, I would like to turn to the next step of our webinar tonight and introduce David Page. Our speaker, David Page, is the CHS Coagulation Safety and Supply Consultant, but a long-term member of the Canadian Hemophilia Society, and working closely with the members of the Blood Safety and Supply Committee for the past 30 years. And the BSSC committee works to rigorously monitor the latest scientific publications and communicate important issues within the bleeding disorders community. So David's diligent work in relation to products and development is more than essential to our understanding of the advancement of gene therapy. And with that, I will turn this over to David. And thank you very much, David, for your presentation tonight.
Thank you, Sarah. And thanks, Roy, for that great presentation. So I'll be quite a bit shorter. I'm really presenting on when and if these products are really coming to Canada. As Dr. KHALIFÉ said, they're licensed, but they're not yet available. And so I've called my talk one step back and two steps forward, which is somewhat optimistic. So I'm not a doctor, no recommendations, and most of these opinions, some are mine, but they also reflect, I think, the CHS and its Blood Safety and Supply Committee. Those of you who were with us in the spring for the first of our webinars, which was called A Long and Winding Road, just a reminder that we've been working at this for 40 years. The cloning of the hemophilia A and B genes was more than 40 years ago. And it's taken that long to get the first hemophilia gene therapy approved, first in US and Europe, and now in Canada. So again, a reminder on who makes the decisions. So the first decision is on approval. Who approves these drugs? Are they safe and efficacious? And that's Health Canada's job. And in this case, both Hemgenix and Becvis have been approved by the regulator. That means they are considered to be safe and effective. But are they cost effective? So who makes those recommendations? Who makes recommendations to the public drug plans? Well, we have two HTA bodies, INES in Quebec and Canada's Drug Agency, formerly called CADIF, who make those recommendations to governments. But they don't make a decision. And in this case, for gene therapies, they then go to a price negotiation. And that price negotiation is conducted by the Pan-Canadian Provincial Pharmaceutical Alliance on behalf of all the public drug plans. And so then again, they come up with a price, and then it's up to the provincial territorial ministries of health to make a decision on whether or not to reimburse the drug, to pay for the drug in the public sphere. And unfortunately, I think in this case, it looks like we'll have 13 different decisions. There'll be one price, but then it'll be up to each province and each territory in the country to make a decision on whether or not to reimburse it for their citizens. And then finally, even if it is reimbursed, there comes the shared decision-making process with the physician and the patient and family. So the one step back, you've heard about that Roctavian is not up for approval here in Canada, even though it was approved in August 2022 in Europe and June 2023 in the States. But just recently, BioMarin announced that it's limiting marketing to only 3 countries, US, Germany, and Italy, where it's currently being reimbursed and prescribed. Last I heard, you could count the number of people who received Roctavian on a commercial basis on one hand. It may now be 2 hands. So I think we're talking about somewhere under 10 people have been treated commercially with this product. And that, I think, is one of the things that's led to BioMarin concentrating their efforts on those 3 countries which are already actively commercializing the product. But the 2 steps forward are really related to hemophilia B. So Hemgenix and Becfez were both approved by Health Canada last year at the end, just before Christmas. And since our last talk, CADIF has given positive reimbursement recommendations for both Becfez and Hemgenix with conditions. That's the good news. That's a step forward. But the one back is INES in Quebec has given negative reimbursement recommendations for both Becfez and Hemgenix. That means they're not recommending to Quebec government that this should be reimbursed. It's strange. They made these positive and negative decisions based on the same information, I think with the same representations from experts, and they came to exactly the opposite opinions. Obviously, I agree with the CADIF one, and I don't agree with the INES one. So what are the reimbursement conditions from CDA for the rest of Canada? As you heard, certain medical indications, adults, severe and moderately severe, no history of inhibitors, and then the AAV antibody considerations. In the case of Hemgenix, an antibody level of less than 1 to 900, prescribed through our hemophilia treatment centers. And then the big one, of course, is the price reduction. The price listed in the submissions was $4.7 million. And I can tell you, it won't get reimbursed at that level. What the negotiation will end up, I cannot tell, but it's going to have to be much less than that to be considered. And also have to budget impact considerations. Are there going to be some alternative reimbursement models that will permit that price to be spread out over several years? That was for Hemgenix, but for Becfez, these are the negative. These are INES's negative recommendations and the reasons for it. They say it's safe and effective, but they say the clinical studies have been too short, and the long-term factor IX expression is uncertain. And that's true because the clinical trials don't last. They don't go out for 10 or 15 years. They say that some patients had to return to prophylaxis, which is also true. And they say that receiving this gene therapy will prevent access to future gene therapies. 60% of patients needed corticosteroids, and the long-term safety is uncertain. So the conclusion of INES was they do not recognize therapeutic value. and that was a unanimous recommendation in Quebec. Hemgenics, the recommendation was a bit different. Yet, again, safe and effective, all the same reasons. Only 6% had to return to factor IX prophylaxis. Only 20% had needed corticosteroids, but they concluded for Hemgenics that they did not recognize the therapeutic value, but it was a majority recommendation, not unanimous. And the minority opinion from their expert group was that it should be up to the person with hemophilia B, after informed decision making, to decide whether or not they were ready to take it. So interestingly, neither of Inés's recommendations considered the cost of these gene therapies or their cost effectiveness. They didn't reject these because they were too expensive. They rejected them. They stopped at the point when they came to a negative opinion on the therapeutic value. Note that CHS and the Quebec chapter, as well as the Quebec clinic directors, have criticized the scientific validity of Inés's recommendations to the Quebec Ministry of Health. And so what we're doing is we're waiting for the release of more data on both Becfez and Hemgenics. So 4-year data for Hemgenics, 3-year data for Becfez, and then hopefully we can make new representations to the ministry in Quebec and reverse this decision. So we do have a positive recommendation in the rest of Canada. Prices are being negotiated. So where do we stand? Well, if you look at other countries, it is reimbursed and accessible in the US, in the UK, Germany, France, Spain, some Scandinavian countries. I know that price negotiations are underway in Ireland. And price negotiations started this summer here in Canada with the PCPA for both Becfez and Hemgenics. I can't tell you where they stand. I know they're progressing. I also can't say how long they'll take. It could be 6 months to 12 months to even longer. That depends on whether they can come to an agreement. Now, CHS did make a submission to the PCPA in favor of access. What we did was we highlighted the long-term health and quality of life benefits of people with hemophilia B. And we also stated that we thought there's a potential for cost savings in the long term at the right price. So if you take 10 years of treatment with prophylactic factor IX and compare it to 10 years to a single treatment of gene therapy, we think they could be comparable and cost effective. And then we also think there's a need for creative payment schemes, which involve annual payments as opposed to a one-time payment, risk sharing between the government and the manufacturer, and payment stop mechanisms. So what happens if it doesn't work, as Dr. KHALIFÉ was talking about. So that's where we stand. Stay tuned. Hopefully there'll be some news early in the new year, and we'll certainly be reporting on that through our website and through our communications. If you want to know more about the gene therapy education program, we keep it updated as soon as something new happens. You can go to our website and find that. And I'll stop there, and I think we'll open the questions to both Dr. KHALIFÉ and to me.
Thank you very much, David, for that update. I'll begin with a question for you, David. We're hearing some different terminology than we're used to in our community. And where does Canadian Blood Services and Hema-Quebec fit into all of this? And why is it different in this particular situation than what patients might be used to?
Well, all coagulation therapies to date, including emicizumab, have gone through the CBS or the Hema-Quebec blood budgets and their approval processes. But for the first time, governments decided that gene therapies were in their own separate space, and given all the number of gene therapies that are coming along, they decided they would send these, if they're approved and reimbursed, to their provincial formularies. So that's unfortunate. It's going to lead potentially to some inequity. If say one province says yes, we will reimburse it, another province says no, and we're already going to face that with Quebec, but we may face it with other provinces as well.
Thank you. This is for Dr. KHALIFÉ. What are some of the most common comments or feedback that you're receiving for patients as they're now starting to look at this as a potential new therapy option? And what have you heard just basically generally from the patient population regarding gene therapy?
I mean, it's a mix of responses, and so there's always people who are very eager and want to jump on board, and I think part of it is you know factor infusion takes a toll on people. Hemophilia B, we don't really have any subcutaneous, well, we don't have any subcutaneous options that are available in the clinics. Um, and so I think some people are very much on board and want to go ahead just primarily to reduce or potentially no longer do the infusions. I think that's the main thing. Some of the concerns are, well, will I be eligible? Is my liver going to be okay? Is my liver going to be ready for gene therapy? And part of that is really starting to work on these things and preparing people, but also doing some of these investigations to see. Some people have had hepatitis, C, as we know, and HIV, and these are not exclusion criteria as long as there is no liver damage or no advanced liver damage, and HIV is controlled. The gene therapy would be would be a potential treatment.
Okay, thank you.
And sorry, I guess the opposite end is some people, it's pretty clear cut. No, do not talk to me about that. I don't want, and that's perfectly fine, right? So it's up to each person to decide what is important for them and what is working for them. And if someone's happy on a once a week factor 9 infusion, that's really, great and doing well, and we want to support that.
Okay, thank you. David, where do you see our evaluation and education going forward, even past this phase of the hemophilia B gene therapy trials, and accessibility and reimbursement? Are there other things that are on the horizon that we're now looking at?
In terms of education, yes, I mean, we'll soon be launching a novel therapies education program. So looking at all of the other, in some cases, wonderful things that are coming along, not just for hemophilia A and B, but also for von Willebrand disease, for Glanzmann thrombocythemia. Really, there's a whole pipeline of new therapies that are really promising. They're early in clinical trials or just entering clinical trials, but really exciting for people who have some of the other bleeding disorders.
Thank you. Dr. KHALIFÉ, are there any studies that show whether or not a person is more likely to develop antibodies to prophylaxis after returning to prophylaxis post-gene therapy treatment?
Um, that's a great question. I mean, most people have not returned to prophylaxis, right? So very few have. I'm not aware that there's been any issues with development of allo antibodies or inhibitors to factor 8 or factor 9. Um, but again, people in clinical trials are very highly selected, right? So will that be an issue moving forward? Probably not. And most of our new products we have, on the adult side, it's rare for us to see inhibitors nowadays, which is a good thing.
And these people had no history of inhibitors in the first
Correct.
place, so they're probably less, not very likely
Yeah.
to develop any inhibitors.
Okay.
What we know is for sure you will have inhibitors or antibodies against AAV because we're giving you an infusion of AAV. Your body's going to produce inhibitors or antibodies against AAV, and that's why it's a one-time, once in a lifetime infusion that is AAV based.
Okay. Well, thank you to you both for your contributions this evening. We greatly appreciate it. And on behalf of the community, this is very important for us to continue education and communication as to where we stand. And please stay tuned as we start to see further developments, especially regarding the accessibility issues that are involved with this therapy. So this concludes our webinar on hemophilia gene therapy. And I'd like to thank both Dr. KHALIFÉ and David for your presentations. Also, a big thank you to our sponsors of the CHS Gene Therapy Education Program, Pfizer Canada and CSL Behring, for their unrestricted grants. We also would like to thank you all for joining us this evening, and for those that are listening to this as a recording through our education program. I would like to extend an invitation to join us in May 1 to 4, 2025 for a rendezvous in Winnipeg. That's going to be the next time our community is all getting together. And on that note, if anyone would like to stay for our French webinar, please stay on the line. So thank you and have a good evening.